Thank you for your interest in our poster entitled Qualification of Veterinary Glucometer Use in Yucatan Miniature Swine, presented at AALAS 2024 in Nashville, Tennessee. My name is Mandy Grimwood, Veterinary Services Coordinator at Altasciences in Columbia, Missouri, and I will give you a brief overview of the work we have conducted. Here at Altasciences, we offer a diabetic swine model to enable research on diabetic treatments and management strategies. When working with this model, handheld glucometers are critical for the point of care blood glucose monitoring for both animal care and research purposes. The model of glucometer historically used at our institution was discontinued, and we sought to qualify a new model of use for diabetic swine. First, we conducted a preliminary trial to determine if the human or veterinary glucometer would be more accurate for use in miniature swine. To do so, we collected blood from a previously implanted vascular access port. Whole blood was analyzed in duplicate on three separate glucometers. Our previous used Alpha Track 2, Alpha Track 3 on both available codes, cat and dog, and human glucometer, the Freestyle Light. The blood was immediately processed and serum was analyzed on our house chemistry analyzer for blood glucose. The preliminary trial identified that the Alpha Track 3 veterinary glucometer would be more closely aligned with the blood glucose levels generated by the chemistry analyzer compared to the human glucometer. The dog and cat codes measured similarly, but ultimately the dog code was chosen for the qualification trial as it intended to pass quality control checks more reliability than the cat code. Blood was collected in the same way for the qualification trial. The International Organization for Standardization, ISO, performance standards for glucometer accuracy were utilized to determine if the Alpha Track 3 would be suitable for use. 6 non diabetic and 6 diabetic Yucatan miniature swine were utilized for this trial. The Alpha Track 3 met performance standards for this qualification trial. All non diabetic animals with blood glucose levels less than 100 milligrams per deciliter measured within plus or minus 15 milligrams per deciliter with the chemistry analyzer. In addition, all diabetic animals with the blood glucose values greater than 100 milligrams per deciliter measured within plus or minus 15% of the values generated by the chemistry analyzer. Thus, all pigs fell within the ISO standards. In conclusion, the Alpha Track 3, a veterinary glucometer, was qualified for use to monitor the blood glucose values in our diabetic and non diabetic Yucatan swine. This device allows for fast and efficient blood glucose measurements to aid in diagnostic and clinical care decisions, as well as to monitor the day-to-day diabetic state of the animal. It is our hope that we can help contribute to the better treatment of management strategies for diabetic patients around the world. Thank you for listening.